Hello, my name is Joshua Mugabe. I welcome you to Vision in Insects. We are going to look at vision in insects. The compound eyes consists of thousands of identical units called ommatidia, packed together on each side of the head. So let's look at our compound eye here. This is our compound eye. It has been cut through, cross-sectioned, and we want to see the different parts of the compound eye. We have the cuticle. The cuticle is this outer layer. The cuticle is the outer layer, and we are saying that the cuticle here is transparent and forms a series of lenses. So what we are seeing here, this is a lens, and here we have another lens. All these ones that we are seeing on the cuticle, all those ones are lenses, and those lenses are found on the cuticle, the upper layer, which is covering the compound eye, is what we are calling the cuticle, and it is transparent with a series of lenses. So when you move from this layer here, this upper layer here, which is having the lenses, then we go to the second layer. If we move to the second layer, we have the second layer here. Now these ones that we are seeing here, these are also cones. They are lenses, as we shall see them later on. Then they will now attach themselves. The whole of this structure is what we are calling the ommatidia. From here, if you cut out one lens like this one here, you cut this one here, the cuticle. From the cuticle you come to the cone, you go down like this, and then from also here you go down like this. The whole of that section is what we call the ommatidia. The upper section here is what we have called the cuticle, or which is made up of very many lenses. And then we have here, these ones are the cones inside here. We shall see them later on. And then we have the whole structure here, the ommatidium. And here we have the nerve fibers. So let us try to look at the ommatidium. This one is our ommatidium, the whole of it. And it is this one, if you look at this one from up to down. Up we said we have the cuticle, cuticular lens, this one here on top. It is the one that we are having here. This is the cuticular lens. This one here, it is this one here. Then from there, we went to the second layer. The second layer here, we had the cones, crystalline cones. This one, which we are seeing here, it is the same as this one here. It is the second layer, which we are referring to as the crystalline cone, and it is also a lens with the cuticular lens. Then from the cone here, now we have these ones here, which we are calling the matrix cell. They are the ones that produce or secrete the crystalline cone. Now let us check here if we can see the matrix cells. The matrix cells here, the matrix cells are these ones here. We have on the sides here of the cone. This one is a matrix cell. If you look here, this is a matrix cell. All these ones are matrix cells. They are the ones that secrete the crystalline cone or our lens. So now we can see. We have said that this one up is the lenticular lens. This layer up we have is the lenticular lens, which we are having on top here. the lenticular lens. This one, the lenticular lens. Then when you come down here, this is our cone, crystalline cone, which is here on this one here. These are our cones. Then when we move away from the cones here, we have seen the matrix cells. These are the ones that secrete the crystalline cones. We have already seen them here. Now we come to this part here. This one's called rhabdome. Rhabdome, this tube here. When you come this side, it is this tube that you are seeing in the middle. This tube is what we are calling the rhabdome. Rhabdome. It is a transparent rod. It is a transparent rod, and it is surrounded by the light sensitive cells. These are the light sensitive cells, and this side, these ones here are our light sensitive cells on the sides. Those are light sensitive cells. If you look at different cells, those ones are the light sensitive cells. And in the middle, it is where we are having our rhabdome. This one here in the middle of the light sensitive cells, it's where we are having our rhabdome in the middle there. That is our rhabdome. So here we are having our rhabdome in the middle, and outside we have the sensitive cells. Then at the end we have the nerve fibers. Nerve fibers, these are the nerve fibers that we have here. All these ones are nerve fiber. So now I think you are very familiar with the compound eye. It is made up of the first layer, which is the cuticular layer, which is containing lenses. And then when you move from the cuticular layer, you come to the crystalline cones. They are also lenses, the crystalline cones, and these crystalline cones are produced by matrix cells. Those ones on the sides, those cells on the sides are the ones that we are calling the matrix cells, which secrete the crystalline cones. Then when you come in the middle, we have what we are referring to as the rhabdome. Rhabdome, it is a transparent rod. Every ommatidium has got a rhabdome. Then outside the rhabdome, we are going to have our light sensitive cells. And then the light sensitive cells, they are the ones now that end up forming the nerve fibers. So that is the structure of the ommatidium, which is found part of the compound eye. So the cone of light enters the ommatidium. The cone of light enters in the ommatidium. Now what we are looking at, we want to see how light is going to be perceived by the insect. Here we are looking at light. We have our light rays coming in from up. This is part A. Part A, we are saying the cone of light enters into the ommatidium. We have said that this whole part here is the ommatidium, what is made up of the cuticular lens, crystalline cones, linear lens. We have the matrix cells, we have the rhabdome, we have the light sensitive cells and the fibers. So they are the same that we have here. This one up here is our lenticular cells, and then here we have the crystalline cones. From the crystalline cones, you know, we connect to the rhabdome. From the rhabdome, we go to the light sensitive cells, and then we connect to the nerve fibers down here. So we want to see how light is going to pass through this whole process. So at part A here, we are seeing the cone of light entering into the ommatidium. Then at part B, the light is refracted by the cuticular lens and the crystalline cone. That is at part B. Part B here, we have this part here, which is the lenticular lens, and then we have here this part here, which is the crystalline cone. So light is going to move is going to be refracted through the cuticular lens, and then it will also be refracted through the crystalline cone. That is at part B. Then when we come to part C, light is projected onto the rhabdom. That is at part C. Part C here, you see that light is coming from the crystalline cone. And when it comes from the crystalline cone, it will be now projected to the rhabdom. And remember, we said the rhabdom is a transparent rod. So it is projected to the rhabdom. That is part B, part C. So here on part D, light now is going to travel down the rhabdom. Light is going to travel down the rhabdom. So the light being projected, which is coming from the cone, projected into the rhabdom, it will travel down the rhabdom, which is our transparent rod. Now, light stimulates the light-sensitive cells. This light, which is passing through the rhabdom, is going to stimulate the light-sensitive cells. It will stimulate the light-sensitive cells. These ones are our light-sensitive cells. They are going to be stimulated by light which is coming from the rhabdom. And then the light-sensitive cells, they will fire impulses along the nerve fiber to the brain. So now when the light in the rhabdom is passing through downwards, it is going to stimulate the light-sensitive cells. And when the light-sensitive cells are stimulated, they are going to fire impulses which are going to be sent to the nerve fibers, and then the nerve fibers will send the impulses to the brain. So that is how light is received by an insect. When light strikes the compound eye, it will be refracted through the reticular cells or the lenticular lens, whereby again it will be refracted through the crystalline cone lens, and then it will be projected to the rhabda. And when it is projected to the rhabda, it will move through the rhabda. As it is moving through the rhabda, it will stimulate the light-sensitive cells. And when it stimulates the light-sensitive cells, they will fire impulses and pass those impulses into the nerve fibers. The nerve fibers will send the impulses to the brain. So now we want to look at how the insect is going to perceive these different light patterns. The diagram shows the pattern of ommatidia, which would be stimulated by two shapes. So now here we are looking at this one. Let's take this one as our lenticular lens on top of the compound eye. So when you have these cells here and you project light in this shape, you have light which has been projected in a circular form, and then you have another light which has been projected or in form of a square. How is the insect going to perform these ones? So these are the patterns of light falling on the compound eye. So what is going to happen is that when you look up here, this is a circular one, okay? Here we are having a circular pattern, and here we are having a square pattern. So what is going to happen? Let us see what is going to happen. Now, when the patterns are focused, when they fall onto the compound eye, what is going to be perceived is that these cells where the pattern is passing are going to be stimulated. And when they are stimulated, they are the ones now that are going to appear here like this. So wherever the light is passing, that cell is going to be stimulated. So when you look at the ommatidia, you will see that all these ones where the light passed, they are the ones that are being stimulated. And these ones in the middle where the light did not pass, they will not be stimulated. The ommatidia will not be stimulated. So when you also look at this square here, you will see that these parts where the light has passed are the ones that are going to be stimulated. And the ones in the middle whereby the light is not passing there, they are not going to be stimulated. Which means that now our eye cannot differentiate between the two patterns. So the compound eye could not distinguish between the two shapes. It cannot distinguish between the two shapes because whichever cell, ommatidium, that is stimulated, it will form that pattern, which is similar to this other pattern. So hello my friend, thank you very much for participating in vision in insects. Please do like, share and subscribe.